In a shallow coral reef off the coast of Queensland, something is hunting. You would not see it easily. It lives inside a burrow, a tunnel it excavates in the rubite and coral rubble with methodical precision, sealed at the entrance by a door it builds from sand and mucus. The burrow is its fortress and its laboratory. It rarely leaves. It does not need to. What comes to it, it kills, and the killing is unlike anything else in the animal kingdom. The mantis shrimp, when it strikes, moves its club-shaped appendage at 23 meters per second. The acceleration it produces is 10,000 times the force of gravity. The strike is so fast that it cavitates the water around it, meaning the water itself vaporizes momentarily from the pressure, producing a cavitation bubble that collapses with a force and flash of light and heat approaching the temperature of the sun's surface. The prey is hit twice, once by the club, once by the collapsing bubble. The whole event takes to milliseconds, and this is not the strangest thing about the mantis shrimp. The strangest thing is what it SEESA human eye has 3 types of color receptor. We can distinguish roughly 10 million colors. The mantis shrimp has 16 types of color receptor. It can detect wavelengths of ultraviolet and infrared light invisible to us. It can see circularly polarized light, a form of light that no other animal on Earth was known to perceive. It lives in a world of color and information so far beyond our own that researchers who study its visual system have described it as operating in a dimension of perception we do not have the neuroscience to fully understand. We thought we understood the mantis shrimp. Ancient crustacean, violent predator, exotic vision, a curiosity from a coral reef. We were wrong about almost everything that matters. The story of where mantis shrimp actually came from, how they relate to the rest of the crustacean world, what the genome reveals about that impossible visual system, and what we stand to lose as reef systems collapse, has been completely rewritten by ancient DNA, and what the rewriting reveals is not just surprising. It is, in the precise scientific sense of the word, revolutionary. This is what the DNA actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The textbook version of the mantis shrimp placed it firmly within the crustaceans, correctly, and within the crustaceans assigned it to its own order, the Stomatopoda, meaning mouth-foot, a reference to the positioning of its raptorial appendages near the front of the body. The order was considered ancient and well-defined. Stomatopods had a fossil record extending back to the Devonian, roughly 400 million years ago, making them one of the older crustacean lineages. Within the crustaceans, the textbook story placed them as distant relatives of shrimps, crabs, and lobsters, having diverged from the main crustacean lineage very early. Before most of the familiar modern crustacean groups had differentiated, the standard picture was of an isolated lineage, ancient, set apart, a branch that had left the main trunk of crustacean evolution early and gone its own singular way, developing its weapons and its vision in isolation from everything else in the sea. The fossil records seem to support this. Fossil stomatopods from the Carboniferous and Devonian show body plans recognizably close to living mantis shrimp. The raptorial appendages were there. The basic architecture was established, and the continuity of form across 400 million years seemed to confirm what the horseshoe crab story seemed to confirm, that this was an ancient lineage whose body plan had locked in early and persisted. The genomic data broke this picture apart at every joint. The first disruption came from the placement, when large-scale phylogenomic analyses began comparing crustacean genomes across the full breadth of the group, using hundreds to thousands of genes simultaneously. The position of the stomatopods in the crustacean tree kept shifting in ways that unsettled the old consensus. The problem was not that mantis shrimp were in the wrong place. The problem was deeper than that. It turned out that the crustaceans themselves are not what the textbooks described them as, crustaceans. The genomic data revealed over the course of the 2010s are not a natural evolutionary group in the way the textbooks presented them. The group called Crustacea, which contained shrimps, crabs, lobsters, barnacles, copepods, and mantis shrimp among thousands of other forms, does not form a single exclusive lineage. Insects evolved from within the crustacean lineage, making the traditional crustaceans a paraphyletic group, meaning a group that does not include all the descendants of its common ancestor. The insects are, in the strict genomic sense, highly modified crustaceans that left the water and conquered the land. This revelation, which emerged from phylogenomic analysis and has been confirmed by multiple independent studies, meant that the traditional placement of mantis shrimp within crustacea required complete reconceptualization. The relevant question was no longer where stomatopods sit within the crustaceans. It was where they sit within the broader pancrustacea, the group that includes all crustaceans and all insects together. When that question was asked with the full genomic dataset, the answer was both clearer and more surprising. Stomatopods appear to represent one of the earliest diverging lineages within the pancrustacean group, branching off before the major radiation that produced both the malacostraceans, the large crustaceans including crabs and lobsters, and the lineage that eventually produced insects. The mantis shrimp is not simply an isolated crustacean branch. It is something closer to a deep root of the entire pancrustacean tree, an animal whose lineage diverged before the ancestors of all modern insects and all modern higher crustaceans had separated from one another. The implications of this are profound. The mantis shrimp's body plan, its raptorial appendages, its extraordinary visual system, its unique nervous architecture, all of these features were established in an ancestor that predates the split between the lineage that produced dragonflies and honeybees on one side and the lineage that produced crabs and lobsters on the other. The mantis shrimp is not a cousin of these animals. It is something closer to a very old uncle. The second revelation from the genome concerned the visual system, and it is the one that most completely overturned scientific understanding. The mantis shrimp has 16 types of photoreceptor. When this was first discovered, the assumption was straightforward. More photoreceptor types means better color discrimination. A mantis shrimp should be able to distinguish colors with extraordinary precision, far beyond what any vertebrate can perceive. Researchers spent years designing behavioral experiments to test this assumption. The experiments revealed something that nobody expected. Mantis shrimp are terrible at distinguishing between similar colors. In behavioral tests using carefully controlled wavelengths of light, the animals that should have had the finest color discrimination of any creature on Earth could not reliably distinguish between colors that a human with 3 photoreceptor types would find obviously different. This made no sense, unless the visual system was not doing what anyone had assumed it was doing. The genomic and neurological work that followed produced a realization that has fundamentally changed how researchers think about the purpose of sensory systems. The mantis shrimp's 16 photoreceptor types are not a color discrimination system. They are a color identification system. The distinction is crucial. Color discrimination works by comparing signals from different receptor types, finding the differences between them, and using those differences to distinguish between similar hues. This is what human vision does. It requires significant neural processing, but allows fine discrimination between similar colors. Color identification works differently. Instead of comparing signals between receptor types, the mantis shrimp's visual system appears to assign a fixed label to each wavelength band independently. Each of the 16 receptor types fires when it detects its specific slice of the spectrum, and the animal recognizes the firing pattern as a specific identifier, the way a barcode reader identifies a product. This requires almost no comparative neural processing. It is extremely fast, and it allows the animal to instantly categorize anything it sees into one of a small number of spectral types, without having to compare or discriminate between fine gradations. The genome told the story of how this system evolved. The opsin genes, the genes encoding the photoreceptor proteins that detect light, exist in an extraordinary expanded family in stomatopods. Gene duplication events have produced 16 functional opsin types from an ancestral toolkit that contain far fewer, each tuned to a slightly different wavelength through accumulated mutations in the light-absorbing chromo for region of the protein. The genomic data showed that different stomatopod species have different numbers and tuning of opsin types, and that the elaboration of the opsin family has continued throughout stomatopod evolutionary history, with ongoing gene duplication and tuning changes that have not stopped. The visual system is not a finished solution. It is an active evolutionary experiment, still in progress, but the most unexpected genomic revelation about stomatopod vision did not concern the opsins. It concerned what the opsins connect to. Researchers examining the neural architecture of the mantis shrimp eye, using genomic tools to map which genes are expressed where in the visual system, found that the signal processing that follows photoreception happens in a way that is profoundly different from vertebrate or insect visual processing. The stomatopod visual system appears to perform most of its computation at the level of the eye itself, in the optic lobes that process retinal signals before they ever reach the brain. The brain receives a highly processed, pre-categorized summary of the visual world rather than raw signals for central processing. This is a fundamentally different architecture from the visual systems of almost every other animal that has been studied, and the genomic evidence suggests it is ancient, a design solution that was established early in stomatopod evolutionary history and has been elaborated and refined across hundreds of millions of years, rather than replaced. The third revelation concerned the weapon, the raptorial appendage of the mantis shrimp. The dactyl club that delivers the strike at 23 meters per second is not simply a hard hammer. The genome revealed it to be one of the most sophisticated composite materials ever characterized, a biological engineering achievement whose properties have driven significant interest from materials scientists and defense researchers. The club is built in layers. The outer surface is a highly mineralized impact region made of hydroxyapate, the same mineral that makes human bone, but organized in a helicoidal structure that distributes impact energy in 3 dimensions as the strike lands, preventing crack propagation through the material. Behind the impact surface, a periodic region of mineralized fibers arranged in a rotating stacked structure absorbs the shock wave of the strike, preventing the club from destroying itself in the instant of impact. And at the core, a more compliant material acts as a final absorber. The genomic data revealed the genes responsible for laying down this architecture during the development of each new club after a molt. The mineralization genes, the structural protein genes, the timing genes that coordinate when each layer is deposited, all of them show signatures of strong positive selection across stomatopod evolutionary history. The weapon has been refined continuously, driven by the arms race between increasingly powerful strikes and the mechanical challenge of building a club that does not shatter when it hits, and the club faces a specific engineering challenge that the genome illuminated directly. Every time a mantis shrimp molts, it must rebuild its club from scratch. The new club starts soft and vulnerable. The animal hides during this period, unable to defend itself or hunt. The genomic evidence showed that the genes controlling remineralization of the new club after a molt are among the fastest expressed genes in the stomatopod genome, producing a rush of mineral deposition that hardens the club as quickly as biologically possible. The urgency is encoded in the genome's regulatory architecture. The fourth revelation is the one with the longest shadow, and it concerns the reef. Mantis shrimp are obligate coral reef and rubble habitat specialists for most of their life history. Their burrows are built in the coral rubble zones that border reef structures. Their prey consists of the snails, crabs, clams, and small fish that the reef ecosystem produces. They are not peripheral reef inhabitants. They are deeply integrated members of reef communities, and their presence or absence reflects the health of the reef system they inhabit. Coral reefs are collapsing. The bleaching events that have devastated reefs across the tropics since the 1980s, accelerating dramatically in the 2010s and 2020s, have reduced living coral cover across enormous stretches of the Indo-Pacific and Caribbean. The Great Barrier Reef has experienced unprecedented back-to-back bleaching events in recent years. When the coral dies, the complex three-dimensional structure of the reef slowly erodes. The coral rubble that mantis shrimp depend on degrades, and the prey communities that their burrows depend on disappear. The genomic work on stomatopods made the diversity crisis more visible. Just as genomic tools applied to fireflies doubled the estimated species count, genomic tools applied to stomatopods revealed that morphologically similar populations from different reef systems are often genetically distinct species. The global count of described stomatopod species stands at roughly 450. Genomic studies suggest the true number be significantly higher, which means that as individual reef systems are degraded or destroyed, the populations lost may represent not just local extinction events, but the permanent loss of species that science has never described. The animal with the fastest punch on Earth and the most complex visual system ever, characterized as disappearing from ecosystems faster than it has been described. The genome keeps revealing new species. The reef keeps killing them before anyone has even named them. There is something in this story that goes beyond the mantis shrimp itself. The mantis shrimp genome has become a model system for studying the evolution of complex sensory systems, for understanding the genomics of biological materials engineering, and for exploring how gene duplication can produce rapid and dramatic phenotypic change in an ancient lineage. What researchers learn from the mantis shrimp opsin expansion has implications for understanding how eyes themselves evolved across the animal kingdom. What they learn from the club's mineral architecture has implications for engineering impact-resistant materials. What they learn from the positioning of stomatopods at the root of the pancrustacean tree has implications for understanding the deep history of half the animal phyla on Earth. And almost all of that research depends on access to living populations in intact reef systems. The mantis shrimp spent 400 million years building one of the most sophisticated bodies in the animal kingdom, a weapon that does not shatter when it strikes, a visual system that processes the world faster than any other known architecture, an ancient position in the family tree that illuminates the deep history of insects and crustaceans alike. It did all of this from inside a burrow in a coral reef. In the shallow sea off Queensland, in the rubble zone where the coral meets the sand, something is still hunting. The burrow door opens. The appendage is cocked. The water holds the shape of the animal for a fraction of a second before the strike, and then the prey is hit twice, and then the door closes. Outside, the reef bleaches in water that is too warm. The rubble erodes slowly. The prey communities thin. The animal that solved the engineering problem of striking water at 23 meters per second without destroying its own weapon. The animal that sees colors in a dimension of light we cannot perceive, waits inside its burrow for whatever the ocean still sends. The DNA is only beginning to tell us what it knows. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.